Hello learners, today we are breaking down ELISA topic in simplest way possible. So as we know, ELISA is widely used laboratory technique for detecting and quantifying biological substances such as protein, peptides, hormones, antibody, and it is based on antigen antibody interaction and uses enzyme linked detection system to produce a measurable signal, usually a color change. So let's get started. In this we have a series of topic, and in this today's topic we will be discussing about ELISA, its definition, what is the basis of ELISA, on which principle it work on, elements, components, and how it evolved, and what is a journal procedure in ELISA. So first one is the introduction. So ELISA, it's an enzyme-linked immunosorbent assay. It's a plate-based assay technique which is designed for detecting and quantifying the soluble substances such as peptides, proteins, antibodies, and hormones. So in an ELISA, the antigen, that is the target macromolecule, is immobilized on a solid surface, which is a microplate, and then complexed with an antibody that is linked to a reporter enzyme. And detection is accomplished by measuring the activity of the reporter enzyme via incubation with appropriate substrate to produce a measurable product. The most crucial element of an ELISA is highly specific antibody-antigen interaction. So this is the ELISA microplate. So it contains of wells. So this tray will accompany with such transparent wells, and total there are 96 wells. So what is the definition of ELISA? So it's a detection of antigen or antibody in plasma serum using an enzyme-linked chromogenic endpoint detection system. So what is the basis of ELISA? So depending on the particular assay, either its antigen or antibody is immobilized onto a solid phase. This solid phase is normally a polystyrene or either a microwell or bead. So microwell are generally presented in a 96 removal well plates. So we have seen that wells A to H and 1 to 12. So there are total 96 removable well plate, and the sample assay reagents are added sequentially into these wells, and finally the reaction is read. So why it is known as the enzyme-linked immunosorbent assay? Antigen or antibody of interest is adsorbed on the plastic surface, so that is sorbent. Now antigen is recognized by specific antibody, that is immuno. Now this antibody is recognized by secondary antibody, which is the immunopart, which has the enzyme attached to it, so it becomes the enzyme linked. Substrate react with the enzyme to produce the product, which is usually colored. So it gives the color to that substrate which is having the reaction. That's why it is known as the enzyme-linked immunosorbent assay. Now, the principle behind ELISA is, so the principle based on the formation of the antigen antibody complex, which is detected by the chromogenic detection, which by using conjugated secondary antibody, and this conjugated enzyme act on the specific substrate which is called as chromogenic substrate, which generate the color reaction to the product. Elements of ELISA is coating and capture, direct or indirect immobilization of antigen to the surface of polystyrene microplate wells, then the plate blocking, addition of irrelevant proteins or other molecules to cover all unsaturated surface binding site of the microplate wells, then detection, incubation with the antigen specific antibody that affinity bind to the antigen, and lastly the signal measurement, detection of the signal generated via the indirect or the direct. So indirect means the secondary antibody, the secondary tag on that specific antibody. Components of ELISA. So first one is the solid phase. It's a microplate which is made up of polystyrene. Second is adsorption, so the addition of an antigen antibody diluted in buffer. On incubation, it will attach to the solid phase. Immobilization of the reactant in the ELISA, and the reason for its success is adsorption. Then washing, so we will be flooding and emptying of the wells with the buffered solution to separate the bound from the unbound reagent in the ELISA. Next one is the antigen. It's a protein or the carbohydrate that when inserted in the organism, it will produce the antibody, and antibodies can react with the antigen used and therefore can be used to detect the antigen. Next is antibodies. So they are produced in response to the antigenic stimuli. These are mainly protein in the nature. Enzyme, it's a substance that reacts at low concentration as a catalyst to promote a specific reaction. Specific enzymes are commonly used in ELISA with this specific substrate. Then we have enzyme conjugate, an enzyme that is attached irreversibly to protein, usually an antibody. So then is substrate. It's a chemical compound with which an enzyme reacts specifically, and it will produce a signal that is read as a color reaction. Then we have chromophore, a chemical that alters the color as a result of the enzyme interaction with the substrate. Then we use a stop solution that will stop the process, that will stop this action of the enzyme on the substrate. It has the effect of stopping any further change in the color in the ELISA. So it will stop the reaction and there will be no color change. Then we will read it. So measurement of the color produced in the ELISA, it is quantified by using the spectrophotometer reading at the specific wavelength for specific color obtained with particular enzyme or chromophore systems. Then how ELISA evolved. So the first generation was infected cell lysate used as an antigen in ELISA. In the second generation, there is a recombinant antigens were used. In third generation, there were synthetic peptides. And now the fourth generation in which the synthetic peptides and the antibodies are used. So this is the example like if we see the HIV assay diagnostics testing evolution, then you will see that it has started in 1985 and in 2015 we are in the fifth generation of ELISA which we are using, and the window period is decreased. And if you see the specificity comparing with the 1985, it was 95 to 98%, whereas in 2015 when we started the fifth generation ELISA, it was like 95.5%. And similarly, in case of HCV, if you see it has started in 89, and in 2003 we are having the fourth generation of the ELISA in HCV, and now we can detect the core NH3, NS4, NS5, and the window period from 4 to 6 months it decreased to 26 days. Now how we do it? What is the procedure? So the first stage is we have to add the sample in the well, and that is after the dilution. Now incubate at the specified temperature for that specified period of time. This will immobilize the antigen or antibody, react with any complementary antigen or antibody in the sample, and it will form the antigen antibody complex. At the end of the incubation period, the excess sample and the diluent is washed away. Second stage. In this stage, the conjugate is added to the well after washing, incubated at the specified temperature for a specified time. During the incubation period, the conjugate only bind to a specific antigen antibody complex present, and after binding, it will also become the immobilized. At the end of the incubation period, excess conjugate is washed away. Stages, the enzyme part of the conjugate is used. Synthetic dye, which we are calling chromagen, that is a substrate, is added to the well after the conjugate has been washed away, incubated at the specified temperature for the specified time. This color, this allows the change in the color from the colorless to the color in the presence of the enzyme. At the end of the incubation period, the color reaction is stopped, usually by adding the acid, and the OD, that is optical density value of the individual well, is read. And at last, what we do, all ELISA have a set of control which run with the microplate, which run with the test itself. So we have the positive control, we have the negative control. The result of these control samples are used to ensure that the test run performs satisfactorily and to calculate the assay cutoff value. The cutoff value is the OD value, which is used to decide whether the result is positive or negative. So this is the diagrammatic representation. So specific antibody is bound to the well. Then sample contain unknown amount of antigen is added or antibody is added. Now antigen bind with the antibody. If there is the antibody, then the plate is coated with the antigen. So antibody bind with the antigen. Antibody linked to the enzyme is added to detect the antigen, and the substrate is added, and there you can see there is a color change. So this is all for today's session, and I'm waiting for all of you guys to comment on this video, and soon we upload the part 2 in which we will discuss about the types of ELISA, then later on the advances of ELISA, and the last will be the advantages, disadvantages, and the limitation of ELISA. Thank you.